Hi, everyone. Today we got a special seminar and we got our guests, Kathleen from Rockefeller University in New York City. And Kathleen, she's a project manager for a very ambitious, good project called Vertebrates Genome Project, which is aiming to sequence the very high quality reference genomes for all the extant vertebrate species, about like 70,000. Is that correct? It's very ambitious, and this kind of work will develop an important referencing database to support, for example, biomedical research. And also importantly, and also relevant to all of us here, is about conservation research. For example, this research can help you to identify different genetic variants of endangered species to inform better conservation measurements and planning. So then let's welcome Kathleen for the wonderful talk.
Yep, thank you all for signing on or coming in. Really appreciate you guys coming for an atypical seminar hour. So I'm Kathleen here from New York. I work with the Vertebrate Genome Project, where I am the project manager and also chair of sample coordination. So I work in the Vertebrate Genome Laboratory at Rockefeller University. So this is the main hub of data production for the Vertebrate Genomes Project. You can see here just one room of our lab where most of our machines live. So we've got the 4 PacBio sequencers. These are where they produce our long read sequences, our Bionano Sapphire. I'll go into what those things actually are soon. And also the wonderful team that I work with. Up there on the right-hand corner, we've got Eric Jarvis. So he is chair of the VGP. He's also co-director of the Vertebrate Genome Lab. And he started this project, essentially his background is in neurobiology. He is primarily researching vocal language learning in songbird species and the genetic basis of these characteristics. And he was tired of working with poor quality genomes. Him and a number of other scientists who are equally frustrated kind of banded together and decided, let's start creating better reference genomes, figuring out how we can get higher quality. And the Vertebrate Genome Project was born as a way to create those reference genomes for essentially all species, and particularly moving away. You can find better quality for model organisms, but non-model organisms often provide a little bit prove more difficult to find. So overall, though, the goal is to sequence all vertebrate species, all 70,000. It's a fairly ambitious goal that I think we all acknowledge we'd love to achieve, but we'll see. We're starting out with phase 1 though, which is a representative species from each order. So that came out to about 270 or 268 specifically. And these were chosen along 50 million divergence time. So in the phylogenetic tree, 50 million branching. But we've also got phase 2 coming up next. We're aiming to finish phase 1 within the next calendar year, ideally by the end of the summer. We'll see if this is possible, but we're trying hard. There's a possibility we're going to be reorganizing our priorities, and phase 2 might actually kind of look a little bit more like 1,000 endangered species. So we're working with a number of collaborators and a number of funding sources, and it's a very active planning procedure. But according to the original plan, at least phase 2 would be a representative species from each family, followed by a representative species of each genera for phase 3, and finally phase 4, all species. So we work with a ton of people all around the world. We've got a number of partnerships that are kind of more long-standing and contribute on a higher scale to the number of genomes that we've done with this project or plan to do with this project. Like our Tabula Madagascar project, we're working with Steve Quake's lab from Stanford and Patricia Wright's lab at Stony Brook University, where they operate out of Field Center on base in Madagascar. And we're sequencing a few lemur species for them, as well as every single thing that lemur comes in contact with. So all plants, all insects, anything that it eats, anything it touches, basically, a sequence. We've also done a number of COVID projects, so sequencing different species that might have acted as hosts. We've got the Amazumas project, which is with a group down in Brazil at Vail Institute of Technology, and they have an animal rescue center out there on the edge of the Amazon. and we're sequencing basically all the animals that come through their center. The T2T consortium there, that one you maybe saw in the news like a few weeks ago. So with the human genome, this was the group that essentially was responsible for the completion of it. And we have a lot of overlap with their group, and those they're almost all members of the GP as well. Julio, our bioinformatics lead that you saw there on the first page, he was super instrumental in this project, and him and so Adam Philippi leads that group. But essentially, we're trying to figure out how to apply everything that we learned through that project to also do telomere to telomere level genomes for all the species we're doing. And with all these people, we've had a lot of really awesome papers that have already come out. So back in, I guess it was a year ago now, we had a special issue of Nature came out. So this published our flagship paper. It was the first of our 16 genomes were featured in this, as well as a package of, there's about like 15 other papers, some of which were in Nature, some of which were just promoted with this, covering a variety of things, different algorithms for better assembly of these genomes, different discoveries which were enabled by the creation of these genomes, the sequencing of the platypus paper and echidna paper, the vaquita paper. We also had a couple of years ago, our big one with Nature for the bat species. Whoever does covers at Nature really likes puns. But to date, we are 142 completed genomes out of the 268 for phase 1. So these are all 100% completed. They are accessible publicly. One of the big core elements of the Vertebrate Genome Project is open access. So once we've completed a genome, it's uploaded to NCBI and anybody has access to use that genome. There is a data use policy that states an embargo. Essentially, we have rights to first publication on these genomes within a certain time period. And anyone who contributed to that genome, so whether it was someone who helped with the assembly, with the data production, someone who provided the sample, all these folks are considered like owners of that genome, and they also have those same first rights of publication. And alternatively, there's always the opportunity for anyone to contact any one of those people and ask for permission to publish on it prior to the end of that embargo. So we're all about people being able to actually use these genomes. 62 genomes are currently in progress. So in progress can mean anywhere from that sample is sitting in our freezer untouched, all the way to it's actively being curated, which is the final step before publication. So all of these will continue to move over to the completed progress pretty soon. We've also got 36 that are kind of in flux. The yellow category there is anybody that we have identified a sample source with for these targeted species for phase 1. There's a lot of movement between the in progress and the light blue, the yellow, and the red. Those kind of swap around all the time. Like 2 weeks ago, those numbers were very different. It's a very difficult process to actually get not just samples, but samples that can successfully be sequenced. So sometimes it'll make it through, we'll try extraction, and just no good DNA will come out of it. Other times, someone thinks they have it, they go back to their freezer, they don't. Other times, there's a number of barriers that we can get into that block us from ultimately being able to have access to certain samples. And then some are just really difficult to get a sample from in the first place or find someone who has a sample. And then there's those final 5, which are just amphibians exclusively, and they have absolutely massive genomes. And we have at this point decided they will not make the cut for phase 1 because technically speaking, we have sequenced the genome for each order of amphibians. For both amphibians and reptiles, the selected species for phase 1 definitely went beyond just a representative of each order. And they were more representative of each family, more or less. And so we've decided we're okay with where we are to call those in phase 1. And those 5 can wait a little bit longer until we have more manpower and more machines for sequencing power. Like the average bird genome, to give reference, is 1 gigabase, and the Alabama water dog, which is one of those 5, is 138 gigabases. So a genome, I think it was 16 gigabases. It took almost 2 or 3 years to sequence over at Max Planck. So yeah, we decided for the sake of everything. Of the phase 1 species, these are a number that are currently in that either red or mostly red, but some of them may be in the yellow category that are in Australia. If any of you know anyone who's working on these species and would benefit or be interested in having a reference genome produced, we do have funding completely. So that covers everything from like sample collection, permitting, shipment, all of that through sequencing. Just wanted to put that out there if anybody is working on these species or has a colleague who is. So what we're planning to do, we've had a number of papers published, like I said, but there are a number of things planned specifically once we have completed phase 1. One of the biggest and most important is reorganization of the family tree. So we expect that with all of these genomes sequenced at a higher coverage, it's going to really kind of reorganize a number of branches. So like number 4 there, you'll see reconstructing common ancestor genomes of vertebrates. Yeah, we expect that things are going to look kind of different. So also it's going to open up for a number of comparative genomics of specialized traits. Like I said, Eric, our PI, is specifically focused on vocal language learning. There's a couple things in the works there. Differential disease resistance in different lineages. So for example, we're working on the Corbury frog. So that's a frog located down in, we're working with a group down at University of Melbourne for that one. So that's down there, and they are looking at chytrid and the Corbury frog. For those who are not familiar, chytrid wipes out frog species, just like absolutely massive, terrible fungal infection. And some frog species are completely resistant to its effects. Others, it's an absolute death. So like the Corbury frog, that one, it dies if it gets chytrid, but it lives in very close related quarters with another species that's completely resistant to it. And things like, how do they live together? And why is one susceptible to death? Why isn't the other? These can be looked at, and that's the plans for the Corbury frog genome that we're putting together down there. Conservation genomics of endangered species. That, yeah, a lot of these are really, really broad topics that are going to be covered in a number of papers, but we've got a lot planned. So before I go any further, I just wanted to do a brief review of terminology. I know we might have potentially mixed audience and work with genomics. So whole genome sequencing refers to sequencing. When you sequence DNA, there's a number of ways you go about the actual production of that data. NextGen is what we use, which produces long read sequences. So it's just a more contiguous line of DNA. You get different fragments of that. So traditionally, it's been shorter sequences. But to get a whole genome sequence, you take all of those short or long sequence, all of those fragments, and you assemble them together into one long sequence. A reference genome is one of those whole genome sequences, hopefully at the best quality that you can get, to represent the genome of a particular species. So our high quality reference genomes is a config of N50, 1 million base pairs. So for reference, a kind of typical reference genome might have as few as 80,000 base pairs. So ours, that represents a very, very long read sequence that we put as our absolute minimum for that point. With 90% of the genome assembled into chromosomes, confirmed. We try to have everything haplotype phased. So that refers to if we have the parental data, we don't require, because it's not always feasible, but yeah. Some other that will become relevant as we go through: single nucleotide polymorphism, SNPs. So that's common genetic variation among people or among organisms. So this represents a difference in DNA building blocks. And you can use these in different types of studies. It can look like eDNA or really looking at any DNA. It helps you to identify differences amongst individuals. It helps you to identify like that an individual is existing compared to something else when you're taking an eDNA sample, environmental DNA. It can help you identify disease, a number of things, a number of applications, and DNA barcoding. So yeah, like you'd see the barcoding strip on the back of any kind of product. A DNA barcode exists. It's like a massive database. Every time you upload species information, it's categorized here. You can reference this and confirm that the species you're looking at is the species you think it is. So this is the pipeline that we follow. So you'll see this kind of goes through. Like I said, we use long read sequencing. So up here, the long read sequencing, these red boxes, I don't know if there's a mouse here, you can see it, but yeah. So like that red box, if you follow it down through, that represents the same contiguous sequence. So we get the longest piece we can, and then we've got the linked reads. So once we've got scaffolding, so first, the long read is produced with PacBio. This gives us our long reads that we then scaffold together using Hi-C. So Hi-C helps us to identify chromosomes. It helps us to put these sequences into the correct order. I mean, scaffolding kind of carries the same meaning in this application as it does in construction. And optical mapping, which is our Bionano. So Bionano, it helps us to better do this and label the sequences. So once we've actually put everything together and we run it through assembly, this is done through a number of algorithms that are totally over my head as a non-bioinformatician. But this helps us to actually put everything together in an accurate way, and we can start to purge duplications. So when you've got a full line, a full sequence, you're going to have both the mother and the father's DNA, and there's going to be the potential for false duplications when in fact it's actually just an overlap of things. And we can produce our draft assembly and go into curation. So the curation stage right now is done manually, but curation allows us to identify errors. We can see where there are gaps and begin to, with best knowledge, fill them in. We can identify when a sequence has been inverted and fix these inversions. So for example, with our platypus genome that we had published with that Nature issue, we actually identified 18 structural differences and 13 different chromosomes from that which had been previously sequenced. So the other previous existing standard reference genome. So to get through this process, though, we need high quality samples. So the biggest problem we have a lot of the time with samples coming in, it can be really difficult if you are not able to connect with a scientist who is actively going out into the field and collecting, where we can inform them on best protocols. So a lot of the time we also can just kind of take what we can get, because we also want to be conservative in our approach, and we're not trying to sacrifice if it's ever avoidable. And yeah, museum samples, archive samples, always willing to try things, but we've found that there are particular forms that have the best success. So almost across the board, freshly collected blood, flash frozen is the gold standard. That's not always possible. Not everybody has access to liquid nitrogen, especially on the field. Not everybody has access to a reliable -80 freezer. Not everybody has access to dry ice. So there's a number of things that can really cause complications. We found, for example, we have a dry shipper, which is like a container that you can charge with liquid nitrogen that we can send to people that they can take on the field and to ship back with us as one workaround. But yeah, there's not always a way to do that even. We have found sometimes, particularly bird blood is a little bit more resilient. So bird blood treated with ethanol at room temperature, that can be okay. But like mammalian blood is really, really tricky. Amphibians are notoriously difficult, not only because of their genome size, but they are just very difficult to extract from. And yeah, so best practices there are really appreciated. So this is the amount that we need of each kind, but this I always just find interesting. So this is actually DNA there in the tube. So that's extracted high molecular weight DNA. DNA actually is basically it's like hydrophilic, and you can get it to precipitate in a solution there. So it will bind with whatever surface you've put in there. And then you can actually just physically see the DNA, which is fun. So like I've said, our hi-fi sequencing with PacBio, the difference of long reads versus short reads and the importance of using PacBio. The longer the read, the more comprehensive you're going to have in coverage. So we aim for 30x coverage. And when you've got the short reads, you end up with a lot of missing areas, and you get a lot of things that look like they're going in the right place, but yeah, it can be overlapping, they can be missing. It really, really inhibits your interpretation of that genome. The optical mapping, Bionano. So this is where we label things, but it also just runs at a machine and looks really pretty. And sometimes we all just like to sit there and watch it run. Very, very calming. And then our Hi-C. So like I said, this helps us organize along chromosomes. DNA sequences found on the same chromosome tend to be in a closer physical space. This helps us to identify that process. Once we have actually finished data production there, we upload everything. So yeah, we've got everything there available for our assemblers. So our bioinformatics team take it from there. We have everything hosted on Amazon Web Services. Everything is available then. Basically, NCBI hosts everything for us with open access. So we've also, though, I don't think I've mentioned yet, we primarily sequence everything at the Vertebrate Genome Lab, but we also partner with the Sanger Institute and Max Planck Institute in the UK and in Germany. So they sequence a number of species for us as well. And curation and annotation is done over with Sanger and NCBI. So overall, this is all of the vertebrate reference genomes that have been uploaded on onto NCBI. And this is where all of the Vertebrate Genome Project's genomes live. So you can see here, we've got our line for the config N50, our scaffolds for N50, 10 megabase per, 10 million base pairs. And this one up here, actually, where it was an error. This is not ours, but it's like literally a typo, put it up here. So you can see ours are of the kind of top tier, highest coverage. And part of this is because we use trios. So this, like I said, you've got both mother and father DNA when you come into an individual. And so if you're not able to have access to both of their DNA simultaneously, we'll sequence both offspring, mother and father, when we have it available every time. And this is so in the assembly process, this is known as phasing. So this allows us to separate manually and look at the haplotypes on each chromosome. But this helps us to reduce false duplications and increase continuity. So we've got the offspring, we've got their maternal, their paternal, father's DNA, the mother's DNA, and once we're able to phase it, you can actually separate out what's coming from which. And this, you can see just how much it decreases the duplications there. So down from 5 to 1.4, and we can get even lower. So it's not always feasible to get a trio of the offspring and the parents, but anytime it is, it's going to get you a better genome. And finally, where does this all actually apply to conservation? So when you have a really high quality reference genome with the absolute maximum coverage, it's going to give you a lot more reliability and what we're using those reference genomes for. So the reference genome exists in the first place as a way to kind of, to use as a reference. So you've got anything that you're collecting from eDNA, for example. So we're working with a group called African Parks. They have 13 protected areas in Africa, and they monitor a bunch of species down there. And they're constantly looking for better ways to keep track of the populations of animals. for example, there is a dugong population off the coast that they're monitoring where there's, I think they might be down to just one female. There's less than 200 in that particular population that they're looking at. So it's at a very delicate position. And being able to use environmental DNA, which doesn't involve actually interacting with these species, because even trying to go and interact with any of them can be very, very disruptive to the animal. It can traumatize the animal. So anything that's non-invasive is going to really help you if you're worried about that disruption. But eDNA is, so eDNA you can gather from water, you can gather it from feces, from feathers, from really a variety of sources, soil. And it's a way to monitor wildlife, but it can also be super contaminated, exogenous DNA. It can be difficult to really interpret. And when you don't have a reliable source to refer back to, it can be, it's unreliable in what it is that you're actually looking at. So SNPs, like we said, those are often used to identify what species you're looking at, which individual you're looking at. But if you're working with a fragmented bad reference genome in the first place, it's a lot higher rate of error. Furthermore, detection of cryptic species, which, and having a better reference genome can really, really help differentiate between these things. So also with assessment of population data, with population health, so we had a study we did for the vaquita. So the vaquita, there are 9 left, I think it was at this point. And the fate's not looking good for the vaquita, unfortunately. But what we were able to do in sequencing that reference genome is look back at the population health, like the history of it, and determine that it wasn't, in fact, a health issue in that population that caused its decline. It was human interaction. So while it's like too late for us to really act on that, it is at least a useful tool that we can see there as one application. So you can also evaluate phylogenetic diversity with these. So this can help identifying local adapted variants. And all of these are used as tools for genetic rescue. All of these are tools for guiding management of these species, how to best approach what you're prioritizing in that management. For making assessments on inbreeding deleterious mutations. So for example, we have here, this is the Seychelles magpie robin. We have a woman over at University of Copenhagen who's working on these, and she is looking at the inbreeding in these populations and their population health. She had a reference genome she was working with, and it just simply could not tell her because of how closely related all of the individuals in that population were. She couldn't tell who was a mother, who was a cousin, any of these based on that reference genome. And so she came to us to get a higher quality so that she can actually proceed with her study. So a really great one that we've worked with is the kakapo. With protection and permission from the Gaitahu tribe of the South Island of New Zealand. This one represents kind of a number of things for us. I think it brings in the element, it's a lot more often that it's not just sequencing the genome. These species have importance to people around them, and there's a cultural relevance, there's a meaning beyond just biodiversity. And that means not just, protecting that in the sense of keeping the species alive, but also protecting what that meaning is. So working with a tribe there in New Zealand meant also adapting our data use policy and respecting what they feel they have, they want to protect about this species. So while we do have the agreement for their reference genome, it's still open access. The subsequent other 200 or planned 200 will be protected by a firewall. And basically it kind of illustrated the flexibility that we need and the different levels of understanding that it requires when you work with a variety of sources. So the reason though, this kakapo, they're critically endangered. They're down to, what is it, 200 about there on Stewart Island. You work with kakapo? Oh, okay, yeah. And So this project, we worked with Neil Gemmel's lab down at University of Otago, and essentially we were able to look at the population data that they're collecting using primarily eDNA, but they're holding it up against our reference genome, and they've been able to show kind of what was not expected, but was at least positive in that the species is looking better than we would expect, which let's go into here. So this here is showing what's called a primary component analysis, which is, but simply is just an analysis of difference. So we've got population that was historically here on the South Island, and they took, I believe it was 43 museum samples. So museum samples are really, they don't produce good quality DNA. They're super fragmented, but they can still be useful when you're holding it against, better genomes. And then they've got the live population on Stewart Island as well. So they were able to show that there's actually much more genetic diversity in the historic population here on the mainland compared to the Stewart Island population, which is actually very genetically similar. So that shows a lot of inbreeding. But contrary to what we understand, typically inbreeding would result in recessive traits becoming more and more frequent, a deletion of the greater dominant traits that typically promote the health of a population. And despite the fact that, there's been massive inbreeding, they've actually had pretty low deleterious genetic load. So they don't have as many of these traits that promote poor health in a population. So despite the fact that they're a really very small and circular family tree, they're actually pretty healthy. So we've also been able to see that you can look over population health historically. So we sequenced a number of individuals that you can see here years in the past. So this over here is present, and this goes back a very long time. And you can see each of these blue lines represents an ice age. So there was a drop in population after each ice age. And it's kind of assumed that at some point during one of these ice ages, the population from the mainland was able to cross over to Stewart Island. For those of you maybe don't know that the kakapo is a flightless bird, so it couldn't just fly over, and can't still. So anyways, that contained these to the Stewart Island population. There is, it was helping to confirm, though, we see that pattern in all of the same species that each of these lines is a different individual. So hope is not gone for the kakapo is the simplest way. And they have really valued the use of the high quality reference genome that we produced for them, and credit a lot of the success of this study to being able to have that. So full disclosure, this just got thrown out of completely in the wrong order at some point, but this is a slide showing when you have a trio and when you do not have a trio. So this is meant to show you when you've got the mother and the father DNA as opposed to not. So all of these here without mother and father, they can't be lined up to show us the separation between where that DNA is coming from and where it should be aligned, which chromosome it falls into, how to haplotype phase. So essentially you can say, like, you can see what looks like the mom's DNA, what looks like the dad's DNA, like these two chromosomes, these two chunks look the same, separate them out. Mom's DNA looks like the child's, we'll keep it together. But yeah, sorry, that was meant to be with a different section. So challenges and roadblocks. This one, it can be really difficult to get samples around the world, like I said. Sometimes it's just as simple as having access to dry ice. Sometimes it's about permits, sometimes it's about really a lot of bureaucratic barriers. This was there was a sample we were getting from the Baltimore Aquarium. My family happens to live in Baltimore, and it was the easiest way at the time. This was when, well, still things are disrupted, but supply chains were especially disrupted by COVID. And I had to go myself on Thanksgiving and go pick up from the aquarium with our dry shipper some shark blood. Because at that time we'd lost almost 2 separate shipments because FedEx kept getting them like held up in warehouses, and they're just there melting on dry ice. And we had like a week prior gotten a bag that was so it was like full of gas, and it was just, oh, it was terrible. So we gave up on FedEx for a little while and went very direct. But so access to samples can be really just very difficult. And when we have museum and archive samples, so that's a lot of the ones we get referred to, but they're often preserved in techniques that are incompatible with protocols. Anything that's in formalin is never, ever going to get you the quality of a reference genome that we need. They're also often just, there's no metadata that has been kept for them. So it'll be something like, you've got a fish with like a sticky note in there that's like collected 1992, and that's the only information you have on it. Bureaucratic barriers really form the longest, like that'll delay something more than anything, which anything from permits, like it can take a very long time to get a collection permit to then subsequently get, a lot of these CITES protected species, you have to get your export permit in line. Depending on where you are though, there's a number of other permits that you need, sometimes as much as like 8 for one species from depending on which country you're coming from. Sometimes you need to get permission from your local government or your local tribes, and these can all cause huge delays. Sometimes it's necessary, sometimes it's just frustrating. Nagoya Protocol also dictates the sharing of genetic information internationally. The US is not a signatory, but we work with loads of countries that are signatories, so it's all about Nagoya compliance as well. And also protection of and integration of indigenous knowledge and ancestral value to these species and specimens. So figuring out how to actually adequately and respectfully integrate that information into this type of work, which is so inherently objective typically, has been a learning process for us. So what needs to be done, we're trying to focus really on species protection, and extinction is obviously something we're all familiar with. It's just like rapid fire. And what we're trying to do to help where we can is capacity building. So we work with a lot of different organizations around the world. And when we can share our resources, we do essentially. So for example, we've got the Africa Biogenome Project. They are sequencing all the genomes of every living thing in Africa. And we work really closely in how they go about the formation of that project, where to route their samples. We help them with quality assessment of these samples. We've been able to get them severe discounts with different suppliers for really all the aspects of the tech. And so things like this we try to do when we can. And anytime that we can share our resources, we're trying to do that, to also try to distribute the labor, to distribute the knowledge. So we've also got an organization here in Australia that we're starting to work with, who they have access to one PacBio sequel, but essentially you need more. You guys need more in Australia in order to do this on ground here, but it can be done. So like I said, we have 4 in our lab, and we're always like constantly over capacity. So we need to figure out around the world how to get the tech everywhere that it needs to be to overcome the barriers of distribution of the actual samples, because we can't necessarily lose 3 years on a sample when there's a population of that species that is actively dying each year. So with that, like a stronger and wider network where we can promote more local action. Also, establishment of digital sequence information policy. There's not great protection, and there's not good established protocol and policy over what happens with the data that is produced. So that also brings in the acknowledgement and integration of different knowledges into the metadata, standardizing metadata, what type of knowledge is important to each genome. And these are all things that we're working on, and all things that we're hoping to hear from everybody on what they think and how to move forward with that. And I'll wrap it up there. This is our lab. We do a lot of Mario Kart tournaments, and they insisted that this be the closing page. So any further questions, that's my email there. There's also a QR code that links you to our website. Jen Balaco is our lab ops lead. She's fantastic. So is our bioinformatics lead, Julio. They're happy to talk to anybody with any and all questions. And that is the end of the presentation. Questions?